We are set. Everyone good to go? Okay, and Jeannie, you can also take your screen to silent, and here we go. Hello, everybody. Welcome to Loop and Learn, the T1D speaker series. And today is an interesting time because I think you all need to understand the issues that we all deal with it, whether you're on a loop or not loop. And it's infusion sets that don't work. And I know anybody who's run any pump has dealt with this, and you can get any emoji you want. It's usually an awful, horrified face. And we have some guests who are actually tackling this problem. So we're welcoming Diatec Diabetes from Memphis, Tennessee. We have John Wilcox, the CEO, Luis Blanco, the CTO, and JC Gray, the CSO. Nice numbers, nice letters. Okay, they're young guys. They all met in college, and I'll introduce them to you in just a moment. As usual, we do our disclaimer, which is that the Loop app is a do-it-yourself closed loop algorithm. And while it may seem obvious, please consult with your healthcare professionals regarding your diabetes management. Please remember that this project is highly experimental and not FDA approved for therapy. Therefore, you take full responsibility for building and running the system, and do so at your own risk. I put this out last week, and I'll do it again. The open diabetes community in Europe has reopened their survey with an interesting twist. Not that they don't believe everyone's input, but they want some validation from healthcare professionals as well. So they've added to their survey. So if you took the original survey, if you could please just log back in, and what they're going to ask you to do is request some feedback from your healthcare professional, and you give them permission to do that. They are gathering fabulous data, and if you can join them and help them out, that would be great. And as always, we tell you about loopandlearn.org. It is a rather amazing website, and the more I see things in it, the more I'm amazed. And you will notice that a newsletter went out just about an hour ago about kind of an urgent update that you all need to be aware of if you're running Night Scout. So please bookmark this website. It will tell you things on how to start Loop, how to deal with problems and issues, any announcements. And today is an example of one of the announcements. And then all the announcements of presentations and other hot topics that we're talking about. Sign up for the newsletter. It's really pretty fabulous. And every week it gets better. I know I'm bragging, but it really does get better. So make sure you bookmark it, and there you go. Alrighty, I'm going to stop sharing. I'm going to introduce our guests. And this is exciting. This is a young startup, and they're based in Memphis, Tennessee. It's Diatec Diabetes, and they're developing the SmartFusion, a system for insulin pumps that monitors insulin delivery for accuracy and infusion set failure. SmartFusion software should be integrated into an insulin pump or app and use data from the pump, continuous glucose monitor, and patient infusion habits to monitor the success of daily infusion dosages. I can take you through why this is important. I can tell you what the problem is. We all live with the problem, which is things fail. And they fail, and you don't find out about them till many, many hours later, and then you play catch up for hours and sometimes up to days trying to get your blood sugar under control. You all know this one. And their solution is SmartFusion. It's a system for pumps that monitor this delivery for accuracy and give you early warning. The team is made up of John Wilcox, who is CEO, raise your hand, and he's also a T1D, and he's probably a lot of the impetus behind this. Luis Blanco is the Chief Technology Officer. JC Gray is the Chief Scientific Officer. Hi, JC. And there's one more, but we don't see him. He is COO. He's operations. He hides in the back. They all met at Florida State University. And what's interesting about this group is that they have a pretty strong advisory panel, including Dr. Ray Havlan from Stanford Endocrinology, who's also a T1D, and Dave Walton, who we heard a few weeks ago from T1D Exchange, and he's also a T1D. we're loaded with people that know what's important, and this one is important. And one of their phrases for Diatec Diabetes is insulin delivery done right. I'm real excited to hear what you have to say. Thank you for being here. I need to make sure you are screen shared and you're good to go.
Okay. Okay, so I'm sharing. Let me know if you can see it.
Yep, we can see it.
Okay, awesome. Thank you so much, Joanne. It's really an honor. I think I speak for the guys when I say that when you reached out for us to give this presentation, it was very unexpected. Because we're such a small startup, we're a small team, we're a young team. So really these opportunities are so great because we really do feel that this is an issue that we should all be talking about, trying to get fixed, and something that we believe, our team believes that can get done. So with that, I want to introduce the company. We are Diatec Diabetes. As you mentioned, we're an early stage startup in a medical device field. And the flow of this presentation is we'll introduce you to the team. We'll go over some of the data that's out there from the industry, trying to educate a little bit, fulfill our obligation to the LoopANDlearn, right? The learning part. And then we'll tell everyone about what we have in the works and what we have planned. So with that said, I want to pass it over to John to introduce himself.
Sure thing. Thanks, Luis, and Joanne as well. This is an awesome opportunity, as Luis said, to meet with you all and learn from you all too, and share about what we're doing at Diatec. So yes, the guy right there in the picture, that's me. I'm John Wilcox. I'm the co-founder and CEO of Diatec Diabetes. Something that I also like to keep in my title is I'm also a patient with type 1. I have been living with this disease for about 16 years now. I've been on a pump for about 10. And just that really has been my calling since getting diagnosed with diabetes, is improving diabetes care and finding people who can help me along with that mission. So a lot of my background is involved in academic research of insulin pump therapy and pediatric endocrinology. And I also do advocacy work mainly within the industry for promoting novel diabetes technology and increasing coverage for that type of technology. But on my non-work time, which seems to be 24/7, but when I get a free chance to do something else on Diatec, I love running, specifically for JDRF fundraisers. I actually did my very first marathon at the end of last year for a JDRF fundraiser. So that's also something that I love to do personally. But yeah, this has been an awesome journey so far, and we're super excited, like I mentioned, to talk to you all today about what we're building. So thanks, Luis.
Thanks, John. Now, JC.
Hey, good evening, everyone. Happy Sunday from wherever you're calling it from. I'm going to start backwards on my slide and say that I'm more of a walker fundraiser for the JDRF. I've done a few of those back before, in the before 4 times when we could get together and walk without any complications. Those are always more my speed. Not to say anything bad about John, I can't rock the short shorts, on a really big presentation like this. So, right, so I'm Chief Scientific Officer. I met the guys I think I was just graduated. I was a year above them, but we had gotten to know each other through, coursework and mutual interested professors looking in all sorts of issues. We actually didn't necessarily link because we were already in a diabetes-focused field or doing anything. John really led a lot of that background investigator work, much when we were not even a company. He really saw the clinical side of things, and of course his own experiences kept him inside diabetes all the time, right? But when I was doing my undergrad, I focused a lot in experimental work. I was a volunteer researcher and student researcher working on a number of different projects. And now today, as CSO, I direct, here I wrote the in silico, in vitro, and in vivo experimental design. So at Diatec, we don't work on just sort of one part of the technology, which is, what's the hardware versus the software, versus, what the clinical, we have to kind of do it all. This is really no different from a lot of the major sort of tier one companies. So it's kind of really exciting to be a team of 4 full-time members, doing doing the simulations with the algorithm, validating it on the benchtop, and then validating it with preclinical and eventually clinical models. That's a lot of what I bring to the table. It's really exciting to work on, and I think you'll see some of that effort during this presentation. I do other things related to general science communication, and I think that it's not related to my job at Diatech, but an interesting area that I'm very focused in is something called Human Practices. This is an organization that's international that focuses on genetic engineering technologies and how to responsibly develop those in tandem with communities and societies that need to benefit from them, but need to understand them. Vaccines are a huge subject, but also open insulin is a huge advocate of this type of work that I do with iGEM, relating, the technology to the people, and what they're doing at the Open Insulin Project, if you know of that, in the Bay Area, is kind of an interesting way where a lot of my interests come together. I really support what they're doing. I support the open source community in diabetes, and I think those projects are extremely cool. So everyone at OpenAPS and Night Scout has the same sort of ethos, and I love that. My background, basic engineering. However, I think I've mentioned enough. Do a lot of different things, a lot of different tech development. Luis, I hope I teed that up well for you to bring us home.
Yeah. Thanks, JC. So I'm Luis. My background is similar to JC's. We were actually in the same program at school. I have a BS in chemical engineering. We both have a major that is chemical and biomedical engineering, which is kind of an interesting sort of a dual program. There's one of the last pictures I took on campus in front of what became my favorite spot my last year, which was the Innovation Hub. They had a bunch of printers, a huge nerd, really got into 3D printing and all sorts of things there. My story with diabetes is kind of I want to say interesting, but I think it's very typical, right? So growing up, my grandma actually had diabetes. She had type 2, but she was insulin dependent. And then my stepdad there in the picture, he's a type 1, had it since he was very young. We're from Cuba, so it's a little different in terms of what's available in terms of resources and education, supplies. So growing up, I saw him go from a syringe and a vial. I never saw the glass syringes, which I've heard about through interviews with users. But then I saw him make the transition to MDI, and that was really interesting, right? He always had his 2 pens on him. I thought that was really cool. And then finally, I think last year or 2 years ago, he finally got on a pump. And it's insane because working in the space, I'd seen the benefits of an insulin pump, and he's on a tandem, and he's like, "Oh my God, this has changed my life. This is the best thing that has ever happened. I wish I would have gone on it before." And I told him that, hey, I'm meeting with some of the representatives. He's like, "Oh, thank them, tell them thank you, thank the janitor, thank everybody there for such amazing technology." And it's really show, how much we've advanced in the field. To give some background, I met the guys in school, as Joanne said, and as they said, I was working on a thesis in microfluidics. So there are what ended up in my senior thesis. We were working on cell capture for synthetic biology, and I was really interested in fluidics, and that's kind of what led me to the team. Outside of school, I learned how to snowboard. Saw snow for the first time like 2 years ago. That's me in Alaska, which is one of the best things that's ever happened. Never thought I'd make it out there. I did 2 years at Johnson & Johnson, working in the cardiology space. That's actually from a picture from a patient with ventricular tachycardia that we helped cure. You can see a large infarct on the left and kind of where we ablated. And then on the right is me going skydiving. And that's a solo jump. Really great experience. If you haven't done it, I highly recommend it. So we have a lot of different backgrounds in the team, and the guys have let me put this presentation together and walk you through what is basically our story and how we got up to this point, and what's the sort of the state of the industry, right? What I'll be saying is opinions, right? It's our personal opinion of what is happening, how we can improve it, and we'll mention some studies and some other companies, and I want to make sure that we remember this is sort of our team and how we've interpreted what's out there. As Jen mentioned, we met in school. It was actually a little bit of dumb luck. We were all inadvertently working in the same lab at that point. John was actually the probably the only person doing something cool. He was working in diabetes. He had a patent by that point. And I'll never forget, I was sitting across the table in a presentation. JC was also there, and then Nick was also with them, and Nick is here on the left. And I'll never forget him saying, he's like, I wear an Omnipod. Where's a pod? And he points to it. He's like, I'm a type 1 diabetic. And the thing I don't like about my pump is that it fails. And, go hours tracing down my blood glucose and not knowing what's wrong. And when I look at it, there's a leak at the site. And I was working microfluidics at that time, and it was like, oh, we could totally do that, right? Young minds, limited, no fear, just saying, oh, we can take that on. And actually ended up being a huge undertaking, right? And when the more we learn, the more we realize that there's still a long way to go. But we're doing our best to contribute. I want to start by saying, a friend of mine recently got diagnosed with type 1. She's 27. She has an A1C of 8.2. She was diagnosed because she was, again, kind of by chance. She had high-ish blood glucose. They did the test, they confirmed type 1. So, when she called me because she said, I know you work in diabetes, so you were the first person I thought of. And I said, that sucks. and there's really no way to really comfort someone like that. But then the next thing I said, trying to, kind of get it together, is it's going to be okay. Right. And I work with enough folks, and I've seen the community and how strong everyone is. And I was like, it's going to be okay. And the main reason is that we have access to so much great technology, right? We have CGMs, we have pumps, we have AAD system. So that was my recommendation: get on a pump, get on a CGM. You get lots of freedom with technology. There's a lot of smart folks, a lot of smart people working on this. So looking at the landscape right now, we have a plethora of options, right? You have Tidepool that's going through regulatory. We have the Dexcom G7 on the horizon. We have the Abbott Freestyle Libre, the iLut's coming out. There's Bigfoot Biomedical. There's smart pens. There's the pumps. There's even inhaled insulin, something I'm sure a lot of folks never imagined would happen. And then on top of it all, sort of the cherry on top is, we're not waiting, right? The open source community is probably one of the most important in this whole development, because it's really you guys that are driving the innovation, right? You're saying we're not okay with where things are at. We want more things. This isn't about making a buck. This isn't about, incremental changes. We want to see real changes, because we're there yet as a society, as a, with technology. Unfortunately, and then I was thinking back to my friend, when I was telling her this, I was like, oh, everything's going to be okay. It's amazing. But things aren't perfect. So there's actually a news headline from last year around February, and it was Medtronic recalls MiniMed insulin pumps for incorrect insulin dosing, right? So one would say the pumps have one job, right? And that's to get medication into the user, into the patient. So this is a huge issue for Medtronic. There was some injury, there was one death, and to their credit, they did do the recall, right? This is the right steps. There's an issue, you recall the device, you try and fix it. But when you look a little closer, and this is kind of why we exist, is that there is still a limitation on these systems to accurately monitor for insulin delivery. And that's sort of surfacing now as we get into more intelligent systems. This article from 2018 from the Associated Press, they did a report they looked at all the incidents from 2008 to 2000 to when the report was published, and they found that insulin pumps have the highest overall number of malfunctions, injury, and death reports in the FDA's medical device database. So what does that really mean? And it means that there's a reporting system for these types of issues, and it's just very prevalent still in the community. There's actually as part of the story, they highlighted an incident where, and this was back in 2012, so outside of when the report was written, but it was an incident where a family had just gotten on an insulin pump, and the daughter had to go to the hospital for a high blood sugar, right? She was hospitalized 4 times. So it's safe to say she was in DKA having to go to the hospital. When they got there, and this is the most shocking part, they told her they said it wasn't the insulin pump, it was my daughter. So it's not the device, it's the person, right? And we find time and time again that people are sort of blame when technology is at fault, right? If your phone can't send a text message, you don't say, oh, it's your fault, your text message didn't reach me. You say, oh, it was the phone's problem, right? The technology. Further on the article, they say they blame user error for most reported problems, noting that the pumps are complicated devices requiring special training for patients. In this community, right, in the open source community, we know that people can learn. We know that they can do amazing things. So to say it's too hard, it's too complex, to me, that's not good enough. So what does the industry look like today for tackling this types of failure? Well, we have about over 1 million pump users worldwide. This number is actually underreported. I'm sure it's much larger. It's a huge industry, billion-dollar industry. It's a growing market. But still, we see that over 80% of patients have experienced some form of infusion malfunction, right? This can be an occlusion, a leak, a kink, all sorts of issues. If these issues aren't caught, right, they can lead to high blood sugar, and that can degrade into DKA. There was a study done in 2020 that tracked how many patients were coming into the hospital for DKA, and they asked them, why are you here? And they found that 55% of them was because of an infusion failure. So it kind of makes sense, right? If you think about it, well, we can't, if I don't know that I'm not getting my medication and I'm going into hyperglycemia, that can slowly turn into DKA because I think that my pump is working. I'm going to keep bolusing, I keep titrating my basal up, but I'm not getting anything. And the pump won't tell me, oh, there's an issue. It's sort of, you think, oh, everything's working, I should be out of the woods soon. No one wants to go to the hospital. It's very expensive. Here in the US, it can be very expensive to just go to the ER visit, let alone get hospitalized. And when folks have these kinds of issues, they get discouraged, right? They want things to work. They don't want to be thinking about, I'm sick, I'm in hyperglycemia, I have to deal with that stuff. So it discourages continued pump use, and they're more likely to get off for another type of therapy. This is known as a pump vacation. So here are some pictures of what a failure might look like. There's blood in the cannula on the first one. There's leaking insulin. This is actually John's pod, and he'll send you these pictures and say, hey, here's another one. I just caught it because it happened to be wearing a shirt, happened to be paying attention, was in front of a Zoom call, so I can kind of look back, and there's actually a mother of a daughter with an insulin pump, and this is, I would say this is probably one of the kind of extreme conditions where someone would go in and check on their child, but at the same time, you want to make sure that they're safe. You want to make sure that everything's working. And sometimes the easiest way to know this is to just go and wake up multiple times a night and making sure that things are working as they should, right? So what happens if we can't catch these failures? Is it just hyperglycemia? Is it just high blood sugar that we're dealing with? Well, not exactly. So infusion failures can be fatal. They actually found in 2018, there was a study published with a trained forensic examiners to look for evidence of infusion set failures in patients that had been recently deceased that were on insulin pump technology. If you see at this figure, here's a picture of a kink. You can barely make it out, but you can see sort of where the tubing is bent and just laying on top. It didn't penetrate the tissue, and the pump was just working, right? And when they look further into the case, they say that the patient had excellent adherence to monitoring and competent adherence to dosing. So they did everything they could. They did everything right. But unfortunately, there was a severe complication, and the last 5 readings recorded a blood glucose of over 400 milligrams per deciliter. As part of that series, there was actually 6 individual cases that they found, and this one was a little similar story. This one seemed to have penetrated the tissue, but became dislodged. And when listed under how injury occurred, they put the instead of insulin pump catheter not secured in subcutaneous space. So it wasn't inside, they were just delivering into basically outside of the body. So what are the types of injections, right? And this is basically a very simplistic overview, but I feel like it's worth looking into, right? You have intramuscular, we're going down in the muscle layer. If you've gotten a vaccine recently, that's kind of where they shoot for, just straight down. The subcutaneous fat, that's our sweet spot, right? That's where we want to go. There's intravenous that happens in the dermis, and then there's intradermal, right? That's in the epidermis. There's solutions that are coming out. They're looking at, can we deliver insulin into the intradermal layer? But this is kind of what we're going for. So when you get diagnosed and when you come on a pump, they say, oh, you have all these options, right, for sites. And you'll see this being published, and you'll see this on the FDA's website, or even, who you talk to, like, you can do all your abdomen, you can do your arms, you can do your legs, lower back, your upper buttocks, and they give you all these patterns, right, that you can follow for proper rotation. And that's really great, right? The more education you have, the better the outcomes. So it's basically simple, right? If you want a successful infusion, we go into the subcutaneous fat. We place our cannula there. We have low A1Cs. We have high TIR. Everyone's happy. We're good, right? Problem solved. Easy. Just make sure you hit the sub-Q every time. Well, not necessarily, right? Because we know that, in our attempts to do that, we can have a leak, right? Which is very prevalent. Like I mentioned, John, that's actually what he came to us with. He's like, I'm having leaks, and a leak can happen due to a number of things. Liquid is looking for the path of least resistance. So if, sometimes it's just right up the cannula out into the, onto the skin. Sometimes it can be through any port. There's lots of different instances of why a leak can develop. There's a kink or an occlusion. The research is still not all there as to why we have kinks, why we have occlusions. We think, and this is sort of something we'll get into towards the end, is that you can have instances of occlusion, or what we call partial occlusions, if the cannula is too close to some kind of layer, right? So if you have the port in contact with something, you could be having some kind of obstruction, right? But it might go unnoticed if for some reason it kind of comes off of that layer, and you have this variable performance. And people will say, oh, I have bad absorption at the site. But really is that they're having some kind of partial occlusion outside of other things that can happen.
Hey, Luis, if you may, not to interrupt your flow, but I'm seeing like testimonials happening in the chat right now about not knowing that you could report these things. So yes, you could absolutely report things. You can even look on the FDA's website these specific types of failures, how often they get reported, and a little bit of detail about what that patient, who's anonymous on the database, but what they experience. And so I actually am going to share that link with you. It's completely open to the public, and it is, I think I'm able to share it like this. There it is. So this is the FDA's database. This is their user-reported malfunctions database, and I believe it's on. Thank you, Joanne, for further blasting that. Just searching the product code LZG, which corresponds to insulin pumps, and I just did like a year, and you could search any period of time you want. You can see which companies are dealing with which types of problems. And it's interesting to note that when you have, say, an infusion set failure, as it might be referred to sometimes, that's not necessarily the manufacturer who's going to be labeled as having this issue if it was to be reported. And if you follow that link, you'll see some pretty bad outcomes, some hospital visits in some cases. And that was only just for the last year, and there are over 10 pages of these reports. And they're not all that bad. Some of them are pretty mild, but they really are all over the place. And so it's interesting that you have this single fault system, the insulin pump, and it has this thing called the occlusion alarm, which we'll get into more, and that this is what's supposed to catch all these events that might occur maybe on the reservoir that you've loaded your insulin into, maybe further downstream, maybe at the site. And it's the insulin pump companies that get saddled with all this, which I think is an interesting dynamic. As you may or may not know, there are many different companies that come together to make an actual product. What you know as your Medtronic or your Omnipod, there are specific contract manufacturers who might make different pieces of it, even things like the adhesive that goes on your body. So when there's issues, it's a little bit of a political game sometimes, who's actually at fault and who actually has to initiate the process with the FDA. So I think that's a little bit of interesting industry background, because when we say that we're tackling infusion set problems, one of the things we have to be kind of not necessarily be totally careful, but we have to be a little bit nuanced. Is, well, whose problem are we solving from the producer side? Are we solving the pump company's problem, or are we solving the infusion set people's problem? Well, we'd like to think that hopefully we're solving your problems in the future, but who takes the technology on and puts it in their system? Yeah, there's a little bit of an interesting kind of interplay between all these people.
Awesome. Thank you, JC. I didn't realize that you could see the chat. I can't see the chat, so this is actually really great. Thanks for jumping in and providing that info. So, as you mentioned, there's really a limitation in being able to pick up on these issues. One of the reasons why we started Diatech, and one of the things we were going after first, was actually damaged tissue, or what we call damaged tissue. So it can be areas of lipohypertrophy, lipodystrophy, scar tissue, anything that doesn't feel right, and you might be injecting into or infusing into. And sometimes this isn't necessarily, they're hard to identify, sometimes without proper education. Sometimes it just feels better, right? It doesn't hurt as much, some people report. So it's kind of a complex dynamic as to how do you identify these sites, how do you prevent these issues from happening, and how do you kind of maneuver if you've been on a pump for 20 years or lifelong diabetic, or a person with diabetes. How do you manage, if all you have are sites with lipohypertrophy, right? And the last one, dislodgement. And we saw dislodgement is something that we don't talk about as much. And you saw that in the fatalities, there was actually instances where the cannula was just sitting on top of the tissue. So it's something that I think we should be discussing more as an industry, and saying, how do we protect from this? And sometimes and people think of this as, oh, I caught my tubing on, my cabinet. It's kind of like catching your earphones, right? It's being ripped out. Or I banged my pod on the door frame, and we know something that happens. There's really no way of detecting that. Right now, the best we have is what we call the occlusion detection, right? And that's pretty much just focused around kinks and occlusion, something that is obstructing the line so much that the pump is saying, hey, I can't deliver, or I'm having trouble. So I'm just going to basically error and let you know. So to spend some time on the issue of tissue health, right? Why is tissue health so important, and what it means for insulin pump users, for folks on multi-day injections. So from a paper published in 2014, they basically found that when you're infusing into areas of LHT, right, lipohypertrophy, and if the AP system continues to increase the insulin infusion to prevent hyperglycemia, so you're having trouble keeping your glucose, railing it in, you're just going to keep infusing into, this may result in a considerable risk that the excess insulin applied induces hypoglycemia later. So going back to my example of, maybe you're next to maybe the muscle layer or some kind of, some kind of structure, right? As you're infusing, you might be getting trickles in because you're kind of bringing it back, right? So you might be kind of partial flow coming out of that site. So you're going to keep bolusing, keep bolusing, keep bolusing. Eventually, let's say it was something in the path and it clears. And all of a sudden, it's like opening a dam, right? So you get all this insulin that's been backed up into your tubing, kind of rushing to the site all at once. And you can experience hypo, after long periods of hyper. In addition, if the next infusion set or injection, if you're using MDI, is applied in a non-LHT area where insulin uptake is faster and more complete. So now you have the opposite problem. So now you're very efficient at getting this medication into the site. The risk of hypoglycemia may be increased, increasing the degree of glycemic variability. So what this basically says is, if you have sort of a preconceived notion of this site or this area works this way, and you go into a site that's all of a sudden performing in excess of what you're accustomed to, you might have a hard time kind of keeping track of reeling in your blood glucose, because you're expecting it to perform in a certain way based on what it's done before, but now you're seeing this new type of performance. So it can be very hard, and the pump doesn't really accommodate for this, right? It just kind of does what it does. And unless you're getting an error code or you're having some kind of a full occlusion, it just says, we're looking good, and let's just kind of work on bringing your blood glucose down or kind of into range. So as part of the lecture, I think it's worth talking about how occlusion detection works, or how the standard is set, how the testing happens for these types of mechanisms, and how we can learn from what we're actually referring to when we say occlusion detection. So from a paper published in 2018, they basically redid the study for following the standard. There's a certain standard that's published out, the IEC 60601, which is basically medical electrical equipment, particular requirements for the safety of infusion pumps and controllers. Basically what lets you build an ambulatory pump and put it in the market. You have to meet the standard. So one of the things they test for is, can your system detect occlusions, right? If it were to fail, right, the pump's job is to pump fluid through the line. If there's an occlusion, can you detect it? So the way they did the experiment, they used a clamp for the Teflon cannula, so the soft tubing, and then for the steel, they just put a rubber cap on it, so it's fully occluded at the tip. They did this for many pumps, right? It was back in 2018. They did Accucheck, they did the Animus, the MiniMed, the Omnipod, the Paradigm, and they tried different types of cannulas, different lengths of tubing, and they have here the maximal time onto occlusion alarm according to the manufacturer, right? So if you take the first one, the first row from Roche, if you're at 1 unit an hour, it'll take you 3.17 hours before the pump says, hey, we're having an occlusion, right? And this is a full occlusion. So we're clamped up if it's a Teflon, and we're kind of capped if it's steel. If you have something like a flow rate of 0.02 units an hour, it can take you 144 hours. That's over 3 days to get that. So if you're wearing your sets for 3 days at a time, you'll just be completely occluded the whole time. You'll never really know. So I have the list on the left of sort of the shorthand version. This is the results from the study on the right. What they found, so you don't have to kind of go through all the charts and stuff, they basically found that only one pump system gave an occlusion alarm after approximately 4 hours. This is the fastest, but most of the other systems took more than 24 hours. It's a big delay. And we're talking, again, full occlusion. This isn't partial. This is on the benchtop. And they're basically just repeating the study that using the standard as sort of guidance. The conclusion from the paper, and this is, I think, worth noting, and it's just taken directly from the abstract, says insulin pump users should not blindly rely on occlusion alarms, but perform regular glucose monitoring, and manufacturers should develop mechanisms that allow an earlier detection at low basal rates, right? So we're basically saying the standard, the way things are running is not good enough to capture what could be very tricky or, infusion environments. I say tricky, but really it's normal life, right? People have everyone uses their pump differently. They pick sites differently. So there should be something more than just saying, hey, we can detect full occlusions. Why do we need to tackle failure detection in education, right? And when I say it, insulin infusion set failure, and JC kind of mentioned this, it's not just about, oh, this is for infusion sets, this is for tube, this is failure. This is really just general infusion monitoring. We need better infusion monitoring. A modern day example of where this might be useful is when you take Control-IQ, for instance, right? So this was given at a public forum in December, or part of the presentation. And what they showcase is how Control-IQ can help individuals manage the diabetes better. So they mention, how they do sleep control, how they do exercise, how they help, kind of get you tighter in range. And we see beautiful data, right? We're in range for almost a full 12 hours, very steady, very, very good management. But from about 1 p.m. to 12 a.m., see a large period of hyperglycemia, right? So it kind of starts after a meal. So you have your postprandial response. You're saying, oh, maybe they under bolus, right? Maybe they forgot. Maybe they actually ate something else and the system didn't pick it up. But then you start to look at the fact the system did correction boluses. There's about 5 correction boluses overall, marked in the top red arrows. And then there's about 5 instances of where the basal rate was titrated up and then brought back down, right? And the glucose response pretty much never budged, except a little dip around 6 p.m. Eventually, we hit a period of 400, on the glucose, and we get a little exclamation part all the way at the bottom. I don't know if you can see, it's right by the lower red arrow, and the pump has finally said, hey, we think there's an issue. Overall, this person was in hypoglycemia from 1:45 p.m. to about 11:45 p.m. after the detection happened, after trying to get it back. So for about 10 hours, they're probably feeling really miserable. There's about 10 units of that came from boluses. There's 5 different events of basal changes. There's a peak blood glucose of 400 milligrams per deciliter, and the alarm finally happened 7 hours after blood glucose failed to come down. So the best we can do is say, we think there was a failure here. And now looking back, we can say this was definitely a failure because they switched their infusion set and it went away, right? It took 10 hours for the system to finally recognize what was going on. And if the user hadn't acted right, they were doing exactly what they've been instructed to, is trust the system, let it do what it's going to do. But they had to intervene, they had to stay on top of it. So this is a real obstruction right now. It's a real sort of obstacle in our way for full automation, right? We're seeing a lot of really positive performance, and from the open source community, we're seeing how changing insulin sensitivity, dialing in your carb ratio, considering protein when you're doing your carb counting, all that's really great. But the matter of fact is, we can't hit full automation is the system itself can't say, hey, I know when there's a delivery issue, and I'm gonna alert the users for it so we can correct and we can just kind of keep going. So where are we right now as an industry? So looking at the last 5 years, we've had BD FlowSmart get announced. We've seen Capillary Biomedical introduce their steady set. Medtronic now has a set that had a heparin filter. I think the commercial product doesn't have a heparin filter. Convatec is launching their Lantern set. They're still in development. This is all great news, except the fact that BD's out. So it was really great technology. BD's out. Okay, we still have 3 more. Capillary's in development. Well, they're a great group of guys. We've met their team. They're amazing. They're actually we were co-featured in an article with them, and we really applaud the work they're doing. We're really excited for it, but still, we really don't know when it's going to come out. The Lantern set's still in development, and as of a couple of days ago, I think the Medtronic set just got EU approved. So that's their extended wear set. And that's really great. But when you think about it, especially when we're talking to a group of mainly Omnipod users, none of this is for the Omnipod, right? So if you're wearing this specific device because you found that this is the best for your lifestyle, for what you want to wear, the pipeline is not there for this type of system. And on top of that, hardware development is expensive and it takes a long time. So I saw Lane Desbro's chat, presentation, and he made a big point about this, that it takes, hundreds of millions of dollars to get through hardware development. It's been a lot invested in very little return in certain instances. So it's very difficult. We applaud everyone for what they're doing in hardware, and we're excited to see what happens. But for now, let's think about maybe a different approach, right? So we've tried the hardware and it's in the pipeline, but what about software? So we'll start here by going over a paper from Sescon. Marja Sescon is actually on our advisory board. She's actually, she's become one of my favorite people over the last year. She's really instrumental to our team. She works very closely with myself and JC on the software development that we do. And she has a healthy dose of, I think, skepticism for when she reviews a paper, especially those in machine learning and algorithm development, because a lot of times things can get put out and very exciting, seems very, very good, but it might just never make it out of research. So when we look through her paper, and this is a publication done in collaboration with Stanford University and Barbara Davis, they had a cohort of patients and they set out the infusion set failure criteria. They said if the meter blood glucose is over 250 and a crushing bolus fails to decrease the glucose by 50 milligrams per deciliter in an hour, that's a failure. If it's over 250 and we see ketones, that's a failure. If there's infection at the infusion site, that's a failure. So kind of laid out their criteria. And what they did is they had these patients, these users, wear the infusion sets for up to 7 days, hoping that there would be an instances of failure, right? Because to protect something, to develop software or anything for a system, we have to study it. So that's exactly what they did. They the patients just used it for 7 days. Here we have a chart of the CGM values from one particular test subject. Around day 6, that's when they had the failure. You could see the graph stops, and they stopped the trial for that individual. We see their insulin intake. So this is when they were, any insulin that they took, it's kind of getting logged. You see that there's, and this might be hard to see, and I'm sorry for not having any indicators for it, but you see there's like 2 little tick marks, and then around day 1, there's a little bit more, day 2 is a little bit more. By the end of it, it's very frequent, the insulin intake that's happening. From this study, they produced an algorithm that was basically able to identify a failure 2.5 days ahead of the actual event as recorded by the clinical team. So this is still having a lot of manual oversight, a lot of personnel investment into saying, oh, this was a failure, this was not. And this would happen, they would call them or they would look over the data. The algorithm they produced was 50%, had a sensitivity of 50%, had an accuracy of 55%, and can basically alert the individual if deployed in real time, a couple of days in advance of this issue. So the goal here was not necessarily to detect it, it was to anticipate it, and it was to capture this degradation of control performance before it negatively impacted the user. And that's something that's really interesting, right? It's not just detection, but it's also prevention, making sure that we don't have to use hyperglycemia as an indicator for when things are going wrong, right? Because nobody wants to be in hyperglycemia. If you look here on the chart, and this is just on the graph, you see that this is where the yellow diamond is when they would make the recommendation, and then the red is when they would have changed. So good work being done by the group, and very interesting paper. You can check it out. Let's look at another example, right? So 2017, Hausman et al. Hausman took a little bit of a different approach. They still use blood glucose to measure to say, well, this is an issue that affects blood glucose. Let's look at blood glucose with the CGMs. So to kind of not walk you through every detail of it, but to kind of give you the high notes, they basically developed what is called they studied the losses in infusion set actuation. So basically they try to identify when things were going bad, right? Or kind of pick up on the pattern that things are getting worse. Develop what they called a glucose fault metric. That basically takes your data, applies some math to it. There's no need to get really into the specifics of it, but basically it says, as your blood glucose is trending up, we can identify and we can sort of say, hey, here's your fault metric, it's getting higher, right? And then they had an insulin fault metric. Same concept here, as you're delivering insulin, if it's not having the desired effect, right, lowering your blood glucose, they're saying, oh, your insulin isn't performing as well, your insulin fault metric. So they combined these two things with the CGM signal, and they were able to produce an algorithm that had 71% sensitivity to these types of issues, right? Issues where they assumed it was infusion set failures. And as part of the study, they would confirm, they would call the patient, they would have them check for if there were ketones, they would have them check the site, and they would try and get confirmation as best as they can when it was noticed that it was a set failure. Here you have the LISA algorithm, which is what Hausman had developed in that team compared to other ones. So you see the sensitivity is pretty much about the same across the board. And they report their median minutes to detection. So with LISA, it was 210 minutes. You do the math, it's about 3 hours, 3 and something hours. So you're kind of riding high for about a couple hours. The issues with this paper, or not the paper, I would say the approach, is not necessarily their fault, is that blood glucose can be very tricky, right? It's a confounding variable. So what makes if you eat a meal, your blood sugar can go up, but if you're sick, it can also go up. If you're on your menstrual cycle, it can go up. There's many things that can impact your blood sugar. Now it's not always up, right? Sometimes it can be down. So you can say, hey, I infused insulin, my blood sugar went down. It's because of the insulin. Sometimes it might be for another factor. There's also alarm fatigue, right? To get this kind of sensitivity, you have to tell people, hey, there's an issue, and they have to hear a constant alarm and making sure and checking, and that can start burning people out very fast. There's also something known as pressure attenuation of CGMs, right? I don't know how much it's talked about, but if you lay on your CGM sometimes at night or in a specific position, you might cause it to have affect its accuracy for the readings that's bringing in, right? And then on top of that, there's a lack of more clinical data sets. Really, this isn't an overly studied problem. There's not, hundreds and hundreds of clinical trials. Just the data sets are pretty limited, and it's a lot of the same times, or I should say, a lot of times it's the same data kind of being passed back and forth between groups trying different approaches. All in all, the biggest issue is that there's no certainty on when failure started, and they require clinician confirmation. So they can say, we think this is a failure, but we have no real idea when it started, right? For the lack of completeness, or I say for the sake of completeness, we'll look at 2021 Meninghetti. So Meninghetti did a different approach, is a more computational approach. They developed the model in silico, right? So synthetic data with a simulation, and they validated on a clinical data set. It was actually the data set from Saskon et al. They basically took that data, right? Time series, they did feature extraction. You can think about it as, if you're pulling features of my face, might be the length of my nose, how wide my mouth is, what color my eyes are. So the features from the data, they ran it through an anomaly algorithm. So basically saying, hey, we have these features, what's the score? Score all these features together and give us a number. And then if it was over a specific threshold, that basically said, hey, we have an anomaly, there's a problem. They tried different techniques, different algorithms, and they basically were able to achieve 75% sensitivity. So about what Hausman was able to do. And they were able to anticipate a set change within 2 hours. If we think back to our tandem slide or case study, we had 10 hours of hyperglycemia. 8 of those were before the detection. So maybe that patient would have had to deal with hyperglycemia for maybe 6 hours or 8 hours, right? Because the detection would have come a little earlier. The limitation here is really what we see in data science a lot of times. You can have a very sophisticated approach, a very well thought out approach, but you always have to think about where we're going. And if this is where to live on an insulin pump, this might take more computation power than an insulin pump has. If it was on your phone, you might have different requirements. The other issue is that it's still using the CGM as a main source of data, right? So if your CGM loses pairing, you don't have that data captured. It's basically just a big blank spot in your data set. So that might negatively affect you. There's also requiring of manual labeling. Labeling is basically when you go in and say, this is this type of event, that's not that type of event. So if you have a group of fruits, your labeling process would be to come through and say, this is a watermelon, this is an apple, this is an orange, and you have some kind of ground truth. In this case, the data is still, and this is basically the big issue in the industry, is we don't have a way to say this is real infusion set failure or a delivery failure, and it happened this time, because we're still relying on blood glucose to do these kinds of labeling, right? So we know there's a problem, but we have no idea when it started, how big the problem is, or why it's a problem. The real issue with blood glucose is that, and this is something that, going back to Lane's talk, he had brought this up, Adam Brown's 42 factors, right? I actually just learned about this recently. I've never seen this table before, this image, and it does a really good job of summarizing what's the problem of using CGM data and blood glucose as sort of the ground truth for your algorithms. If you use that as your ground truth, you're basically saying, this is being affected in the ways that we know. But from this chart, you can see that if you have too little sleep, your blood sugar can go up. If you're stressed, it can go up. If your infusion set issues, you can go up. If you're just smoking, you can go up. If you have caffeine, if you drink, if you have alcohol, it can go down. So it's really all these confounding factors that you can't just say, oh, this is really because there's a failure, right? Maybe you forgot to bolus for your meal, and the system's like, oh, your blood sugar is going up. It's a failure for sure. And so there's something missing. There's a critical piece missing, and that's having some kind of ground truth on the insulin delivery. So one could say we're basically flying blind. And I won't be able to see this in the chat, but if you had to take a guess, random guess, how many sensors would you say an airplane has? if JC, if you see them, if you see a number come through, just shout it out. If not, hopefully we can talk about it at the end. Hopefully Joanne has a number. But if I had to take a random guess, I think maybe 500, maybe a couple hundred, maybe 1,000 sensors or something, right? Maybe they have a sensor per seat, or who knows.
I've got only one estimate by Joanne at 1,200.
At 1,200 sensors. So according to an article in 2017, the new Airbus airliners have 10,000 sensors along its wings alone before it can be qualified to fly. So we're tackling this issue, right, of diabetes, and we really only have a couple sensors. We have your CGM, so your blood sugar, glucose, and whatever the pump is telling us, right? So maybe an occlusion alarm, right? So some kind of occlusion mechanism. So answer is pretty simple, right? Let's just add more sensors, right? We'll add a bunch of sensors, 10,000 sensors per arm, and we're good to go. I don't think anybody wants that. But let's think through what we would have to do, right? And this was proposed by Ignacio Rodriguez in a 2018 paper. So we have a person, right? We have an age, sex, height, weight, body mass index. And it's a person with diabetes. So let's say we're going to have insulin, we're going to have glucose. Okay, that's good. It's kind of the status quo right now. Well, we're going to track your meals. Okay, we kind of do that, in your mind, you kind of have to do that anyways. We'll track your sleep and your schedule. Okay, pretty normal, I guess. The new APS, the new looping algorithm kind of does that when you say sleep mode. What about exercise and heart rate? Okay, maybe we all have smartwatches. Some people do, some people don't. I guess we can track those as well. Why not temperature or perspiration or blood pressure, right? Let's just keep going. So we have all these sensors, we have all this data. We've basically solved diabetes. We can go home, right? We're done. Not really, right? This is a very complex issue. Adding more sensors doesn't give you better data. It just gives you more of it, right? So we need to be very thoughtful about our approach, because there's limitations. There's social, economic limitations. Can someone afford a Fitbit? Does someone even want to track their temperature? Is this too much? People don't want to live with a disease and be reminded that they have to track every aspect of their life. They just want it to work and not have to think about it and go to lunch with their friends. That's an oversimplistic sort of explanation, but that's why we need to solve this and do better. So how can we help, right? Our team has a certain set of skills that we've developed over the years. We're actively developing now, and that's part of our approach to building our advisory board. Joanne mentioned we have Dr. Law, we have Dave, we have Dr. Seskon. And it's really because right now, as we see, so if you think of a patient or a user of an insulin pump, you have your target blood glucose. And this can maybe get lumped in for simplicity with, after you've eaten or before a meal or after you've exercised, you're kind of saying, I have a target that I want to go to. I know kind of what my desired target is. And you have your CGM data and you run the numbers. It might be you, it might be the algorithm that you have on your system. It crunches the numbers and it says, okay, this is the amount of insulin we need. And you have your blood sugar response. What's happening and what's being missed a lot of times is we don't know the health of the tissue. We have no idea if there's a failure. We have no idea what the environment is or the type of site. These are things that we don't track, and tracking them isn't going to make a difference because there's still a big missing factor. And it's that we're analyzing this problem by measuring blood sugar, but realistically an infusion delivery issue should be tracked by the infusion mechanism, right? We should have some kind of indicator for flow. We should have some kind of idea what's going on and the flow. And that's where our system comes in. So we're developing SmartFusion. SmartFusion is an AI-powered infusion monitoring system. We were initially a hardware company. We're developing a smart infusion set. We realized that folks didn't want to have more sensors. They didn't want to pay for more gadgets. So we went more of the software approach with keeping our hardware roots in mind. SmartFusion is an algorithm that integrates into any insulin pump system using data from the delivery mechanism. What this means, if you're an Omnipod, you will hopefully have access to whatever the Omnipod is using to make its decision to know, hey, I'm delivering 1 unit, or I'm confirming them delivering 1 unit. If it's a tandem pump, the same aspect. If it's Medtronic, they have a force sensor, maybe it's a force sensor, maybe it's impedance, maybe it's electrical, but something we tie into that mechanism. We get data from there. And what this does is allow us to generate data for patients and providers to improve infusion diabetes management. If we can say this is an issue, this is a failure, the patient's having a failure, or maybe the converse, right? They're not having a failure and they're still seeing this glycemic variability. We can better troubleshoot what's going on and we can provide better care. We can improve the therapy through the systems that are available to us already. Also being tied into the pump or the CGM or just having information about what's going on, we can better make personalized alerts for folks, right? It can be as simple as, hey, we've detected a leak, or we know you have a lot of leaks at this area, that you infuse a lot, or we're seeing worse performance every time you come back to a spot, try and avoid it, rotate to other sites. It's a very simple, it's actionable data that we hope to give you, and something that lets you just kind of realize there's an issue, have an action, right? So you fix it and then you go on with your day. So if we're going back to our chart or our flow process, so SmartFusion basically exists in a way that it would look out for these types of disturbances. It would say, this is the health of your tissue, this if you're having a failure, if it's environmental, because it's directly tied to the infusion mechanism and it knows what's going on. And it's using that data in combination with your blood glucose and other to give you a ground truth. There's no point in talking about, oh, you have high blood glucose if you can't say this is not an infusion failure. If you can say it's infusion failure, then we just have to change the pod or the site. And then we can start to think about, okay, is it because we missed a meal? Is it because something else? So for the first time, and this is actually our first public disclosure of this data, this is from a recent animal study that we did back last year. This is a preclinical study, so there's still more testing to be done, but basically what we found, and this is using a standard infusion set, we had the standard rates for a pump, and we were basically infusing into porcine tissue, into a swine. Swine have a very close similarity to the type of tissue that we have, so that they're a good analog for the type of study that we do. So in the top left, you see what a normal infusion looks like, right? You have your bolus fluid, you have your bolus field. It looks good, right? You can see the cannula, the fluid going directly to the cannula. You can see it's nice and spread into the subcutaneous. Everything's great. Successful infusion. We've confirmed it with our sensors, with this X-ray. We're good. Now, full occlusion. We see that there's some kind of issue with the delivery, and it kind of looks like that the cannula is kind of being pressed up against something. You kind of see the dip at the center of the image, right? And from going from picture A to picture B, there's a huge difference, right? Our bolus field is much more limited. It's much smaller. Going to a partial occlusion, we're somewhere in the middle. We're not fully occluded, but we're not a normal infusion. And then D, there's a leak and it's all outside the system. It's important to know what you're dealing with at all times, because a full occlusion can lead you down a path of, oh, I might never get this insulin, or I might turn into a partial occlusion. So I might have a delayed response. If you have a leak, you're never getting that insulin, right? It's exited the system. And you're not going to see the benefits of it. We've developed an algorithm that can do 100% classification for these types of issues. We have a proprietary methodology, algorithm, way of calculations, way of doing it that allows us to say, hey, this is normal, this is full occlusion, this is partial, this is a leak. And it all happens within 30 seconds, almost instantaneous, right? We're talking about the papers that say this is going to take a couple hours, might take a full day. We've seen a lot of benefits in what we've been able to do in the lab, and we think there's something here, right? So this is our proof of concept. This is our stake in the ground. This can be done. We just have to think about it in a new way and start thinking about, how is my flow, how is my delivery mechanism impacting? How's the fluid getting into the tissue, and how is that going to impact my blood glucose? So what's the benefit of this? Say we can do it, right? Say we can get 100% accuracy. Say we can say, oh, this is an infusion failure, this is not. The benefit is almost immediate. So here we have an N of 1. So it's a study performed by our one and only John Wilcox. He's very generous and in his time and kind of in what he's kind of put himself through for sort of the benefit of tackling the unknown, right? And what you have here is a very basic controlled study mirroring. It's one protocol. So you take the same, you track your blood sugar every couple minutes. You have the same meal, you have the same occurrence. And in one instance, which is the orange graph, starting at around 0.5, you have an infusion set failure. In his case, it was a leak. So in the orange, you have a delayed response. So didn't know that anything was going on. Crosses the 2-hour mark, 2.5-hour mark, finally notices it, right? You've maybe noticed a wet spot. And you say, hey, this is an issue, I'm gonna change my pod. And you see his blood glucose stays high, and it took a couple hours for it to finally come down. In the blue, you see an instant alert, right? So he's had an issue. He's maybe in a not so distant future, maybe this is what happens. We have SmartFusion, and you can catch it right away. So you still see some kind of a response down the line. Maybe this is the variability we've been talking about, but you're only slightly out of your time and range. He's only hits a little over 200, but he knows anything after that, right? Now he can make sound decisions and say, hey, now I feel comfortable bolusing because I know it's not a failure anymore. So results are almost immediate. You go from a 35% to a 72% in range. You go from having to use 15 units to 8 units, and you reduce the occurrence of DKA. So this could be the world with SmartFusion enabled on one of your devices. So let's say we can do that, right? We've tackled infusion set failure. Remember, we're doing an infusion monitoring platform. Our first sort of goal is to tackle this issue that's been eluding us for such a long time. What happened if we can say this is a failure, this is not a failure? Well, now we can start looking at more complex issues, right? Scar tissue, lipohypertrophy and lipoatrophy, lipodystrophy, things having to do with the health of the tissue. 38% of CSII users place infusion sets on a place they know will work, right? This is common. You've tried a place, you say, I know this is going to work. It might have poor absorption, it might not, but I kind of expect, I know what to expect. But sometimes these skin problems, right, this tissue damage can be hard to detect. And if you kind of pick the wrong spot and you're not on top of it, or it kind of escapes you, you can have a hard time managing your blood glucose. You can be facing reduced insulin absorption. You can have increased A1C, all the things you want to avoid, while treating the diabetes. In a concept, this could basically mean that if SmartFusion is enabled and we know how your sites are performing, we can just kind of give you a really quick overlay and say, like, this is where you've performed well, this is where you've performed poorly. These sites are good, these sites are bad. And we can know objectively, it's not just saying, oh, we've seen your performance where we've worked well. It's objectively evaluating, oh, we know that delivery is good here. And if it's not for whatever reason, we can protect you against those instances that might lead you to this variable performance. Only a couple more slides, but the idea for SmartFusion, as I mentioned, at launch, we're trying to give a true estimation of insulin on board. If we know how much insulin you're getting and how much insulin you're not getting, because there's some kind of impact on your flow path, we can give you a better estimation to your control algorithm. We can protect you from silent occlusions and leaks and give you peace of mind. A lot of folks kind of worry about this, and this is something we're actively trying to understand is, who's affected by this and what is that, what does having these issues mean to different people? And a lot of times we find that it's not that the pump doesn't work or we're having an issue, it's that you don't know and you get blindsided. And you have to suffer all the consequences of just not knowing. And that's what we're trying to tackle. Later on, we're trying to go after tissue dynamics, understanding what makes a successful infusion, how to optimize placement for infusion sets. If you need to change your infusion set for whatever reason. I've seen instances where folks have used a certain type and it's gone really well, and then they lose 30 pounds, right? Because your life changes. And all of a sudden that set in combination with the site isn't working anymore. And the most we can say, oh, I think I have poor absorption. Long-term, the vision is more than a CGM feedback, truly unified AID with error correction and redundancy. SmartFusion is on the ground telling you, hey, we know how much insulin you're getting. We're tracking, making sure that everything's good. Your CGM is telling you, hey, things are happening the way we expect them to. We now have a much tighter AID system and we can really begin to have aggressive therapy management because we know we're protected. So why we need to solve this and do better? We're doing a series. We're actually doing a poster presentation at this year's ADA conference. And I wish we could share the results of the study with you, but we basically did a study last year with T1D Exchange, tied over 1,000 respondents. We have over 700 unique responses to the survey. And we're going to be talking about what are people experiencing, right? How they're managing failures, what's their education like, how does it make it feel, how is this impacting their use of the technology, and how they're dealing with these issues. And this is our part to say is that we think this is a big problem, and we think it's a problem that's not being tackled in the right way, or it's not being tackled at all. And hopefully we have these really, monumental leaps in hardware technology or hardware developments, but we're still not answering the question, can we tell when an infusion delivery is successful, when it's a failure? And we're kind of betting on that the hardware will lead us down a path that we're never going to have to think about this, or we're never going to have to address it. But we think that this is something that we have to actively deal with, and we have to actively tackle, right? We can't continue to build fluid-based systems. We can't continue to build pumps without a mechanism of truly measuring how well it's performing and if the patient is getting it. The pump can tell you, hey, I'm delivering 10 units, but right now I can't tell you the patient received 10 units. We need to fix that. So in pursuit of knowing the truth and what's important to folks, we've actually embarking on an interview series. We have over 40 hours of video and audio from these folks, and it's expressing what they're dealing with, their complications, what's going well, what's going wrong, and what we need to do better. So we've seen a fair amount of folks say, I've left insulin pump technology for MDI because I can't deal with it. I need a pump vacation. I've seen a lot of people say, I wish this existed, and it would allow me to come back to insulin pump technology. So we're really out there. If you want to chat, we're more than happy to talk. We're really excited. We want to learn more about the industry. We want to learn more about the people that rely on this technology and what makes it great. And it's been honestly an honor as someone without diabetes to really get to interact in this level and learn a little more, because the community is so wonderful. It's so amazing. It's so open, and everyone just wants to learn and wants to take on this problem and wants to see these issues get solved for everybody else. Right. So it's honestly a pleasure and an honor. Kind of why we're here today and our ask for the community. So how can we work together with the open source community? This is really sort of open question. Every pump has a sensing element. Can we get data from there? That's really what we're looking for. JC and I have tackled this issue from an external standpoint. We've put on very fancy sensors. We've tried our best to replicate real world conditions, and we're getting the data that says we can do this. We just need a certain level of competency or transparency from the system. So if we can get access to this data, we can start to build our vision of what should happen next, and that's SmartFusion, actively considering the data that comes off these pumps. So this is the last slide. Patients wonder if they are getting the life-saving medication. We must change that. It's John's quote. I think if nothing else, we've done this for him. And I hope you see our passion and everything we've worked towards. And it's really, with the vision that we can tackle this, we can do this. We're a young team, but we're so close to solving this issue that can change a lot of lives. So thank you.
All righty, I'm gonna tell you to unscreen share, and we'll throw a few more questions. JC's been handling them as fast as we can throw them out, but let's see if we can get a view. I'll start with a couple of questions, and I might just throw it out to the group if I can find my chat here. So it's coming from the end to the beginning. Is are these pump companies willing to give you access to that data?
I'll start us off, but I've talked a lot, so hopefully it'd be good to hear from the guys. But we've had some luck. the industry is very open to having meetings and to conversing and to exploring opportunities. Because we're such a small company and we're still in development, there's still a little bit of hesitancy with opening the floodgates, so to speak. But if anybody sees this from one of the corporations, we're here. We're willing to work with you. You have our number. You have our emails. We've talked to you.
Yeah, one of the big things has been being able to share the clinical story. So the story from the actual clinic, the doctors who are seeing this. As you mentioned at the head of the call, Dr. Rehan Lal out of Stanford is on our advisory board. And folks like him mean a lot, I think, to these tier one kind of companies.
Yeah.
And their investigations mean a lot too. And people like Marcia, our data science lead, have explicitly looked at infusion set issues. So we've never met a company that didn't acknowledge it was a thing that they deal with a lot and a problem too. Yeah, as Luis mentioned, I think from the large scale, tech acquisition or huge partnership side, we are not like Diaboloop that you might see, or Type Zero that maybe you've heard of in the news, that are partnered with large pump companies. We are before that, but that's pretty much the model that we're going forward with. So we have some enthusiasm, but nothing we can really share, especially in a public forum. But we hope to, with events like these and more buy-in from the public and from people with diabetes talking about their issues and sharing their stories with us, all of that helps. This platform is extremely helpful for us, but also I hope it's helpful for those watching, understanding some of the nuances that we can share on how new tech actually gets shared and worked on. It's not always the agenda of Medtronic that sort of sets the therapies that you use. It's sometimes a lot more complicated and interconnected than that.
So let me make sure I understand this. Let's take any pump. Let's take Tandem, because it's such a popular pump. It takes readings and it knows exactly. It has a sensor or a recorder that says exactly how much insulin is being delivered or pumped, or the thumb screw moves, or are they able to give you data that says this has pumped this much or this hasn't pumped this much?
So they have to do that by law, is know how much they are delivering into you. But the reality is they need to at least count what they think they're delivering. So these pumps are sort of much more simplistic in these regards than you might think. This is why SmartFusion exists, is because what it's telling you isn't always what's actually happening. To actually measure how much fluid is going into the body, you can't always assume the pump is working exactly the same. Some people in the chat have mentioned things like the temperature might affect whether or not they're having a site failure. The pump simply just can't real test for that. Whenever they're doing their clinical regulation, milestones and things like that with the FDA, these things, they get overlooked or they just can't be implemented. So yeah, you'll see it.
Those things you would have to assume that the person with diabetes has a brain and is aware of what's going on, and it's hot out. But I have no idea how much my pump is actually delivering. It's like taking a capsule of medication, and sometimes it's a little less powder in there and sometimes it's a little more. And that's almost criminal to hear that they're able to give you some range of amount and not be exact on what they're giving you.
I partly think it's a state of the technology and we're not there yet. And I also partly think that we could be there with a few more resources put towards that problem of accuracy.
Okay, I'm going to open up to any of the admins if they want to jump in as well. But there's a part of me that's slightly horrified at this. And I know I actually watched the Asante snap pump being put together by people in the assembly line, and it scared the bejesus out of me because they're just physically gluing things, and it just felt inaccurate, and it didn't feel like there was a lot of tight measurement on it. So are you trying to gather that data that they're putting out, or are you sensing or measuring something?
So in essence, we want to be able to measure directly. So we think that the pumps are underutilizing what they could be doing. And part of that issue is you have to spend some time developing those algorithms. You have this system, such as a Tandem or Medtronic or an Omnipod, and then you've got the CGM companies, and you see a lot of focus on making that open loop and closing the loop, and there are a lot of advances that have happened in recent years, and that's really great. You sort of don't have time to always look at things that maybe weren't really the biggest problem, say 10 years ago, when people were, and there's still many people today, finger pricking and things like that. So we want to be able to do sensing on what the pump actually has to offer. But as you kind of alluded to, every other data stream on that pump, if you're wearing a wearable, like a Fitbit or an Apple Watch, if you have a CGM hooked in, we want to be the algorithm that is sort of the error corrective feature on all of that. So if you are suffering from hyperglycemia and you're not exactly sure if it's from your post-workout meal or if it's from your workout that you just had, well, we want to be able to say, at least your infusion set is working. So you know it's maybe due to some of these other exercise factors. And algorithms of the future are going to take that burden off of you, but they won't know unless they know the insulin is actually getting into the bloodstream. So this is really the core of it, is we want to monitor the actual flow. And for doctors, and I think this is one of the biggest benefits, they can calculate your insulin sensitivity factor without insulin failure or infusion failures being this really pesky issue that they don't really have data on. So what if you're only getting like 75% of your, every unit you get, you're only getting 75% of that? Is the doctor going to say you're more insensitive to insulin and then tell you to use more, and then you're paying more for insulin? When in reality, maybe we can help you pick better sites, pick better infusion sets with different cannulas. As you know, you have many to choose from. Maybe we can help you and your doctor actually use the insulin that you're being prescribed more efficiently.
It seems so obvious. I remember when we found out that finger stick test strips were allowed to have a 20% plus or minus error factor. And at 100, blood sugar of 100, that's 80 to 120. But when you move that range, that's horrifying, and that's very disrespectful to this community. That should be just good enough. I can feel my advocacy things getting louder. How can we as a community interact with you? I can tell you lots of us would be ready for any kind of interviews you want to do. But are there studies? Do you need us to talk to the pump companies? Is it the CGM companies? I mean, this is horrific that there's insulin going in and we don't really know how much. And yet we're measuring on 0.5 units or 0.05 units.
Yeah. I know that this platform, being able to talk about ourselves, and be a little selfish and take the spotlight, is actually incredibly helpful. So for one, I can thank LoopANDlearn and the open source community for letting us talk about ourselves a little bit. But Luis or John, do you have a specific ask? I know there's a lot of kind of techie people in the open source community we'd love to hear from about this issue.
Sure. So Oh, Joanne, I'm sorry, were you going to say something?
Yeah, Sarah just asked, can we send you our
Yeah.
Night Scout reports when we have failures? A lot of us, I don't know if you're familiar with Night Scout, but a lot of us use that, and it gives you a tremendous amount of data, including you can let it know the kind of meals you have or carbs, that you also know what the carbs are. You can talk about activity. And so a lot of data is in that place, and you can actually see, and would that be of interest? Oh, that would be amazing. I see Luis. Okay.
Yeah, that would actually be super awesome. I know there people interact through a platform called Open Humans. I don't know if you guys are on it, but you know, we've thought about how can we engage the community like that and have this data transfer, right? Because we can't tell you this is the exact moment that it happened, but we can open source sort of the ask for data. If you say, I know for a fact I had this incident and the fix was to change my site and it worked, maybe we can start there and say, okay, what can we learn from these instances, these specific, and it basically turns into exactly what they're doing for these studies, but now we have access to a lot more data, a lot more real world data, right? It's not synthetic, it's happening live, and you might have it under all sorts of conditions that we can study.
Well, maybe we should talk, offline about what actually you'd like to see from us, because I can just envision you getting Night Scout reports, a lot of them, and I'm not sure you'd know what to do with those. But maybe a format that makes some sense, that can explain why you're getting that report and what it means and how it's protected so that it's HIPAA compliant, like we really care. Okay, I see. Okay, standard declared. Yeah, we'll talk, and we can put it out there and see if that's useful. I'm thrilled to see that Rayhan's working with you guys. Go ahead. Sorry.
Yeah, no, I first off, I want to say I've just kind of been the fly on the wall for this entire call. So the guys have been doing a great job explaining everything. So Luis and JC, thanks for answering all the questions and as we've gone along with this. But something that I think that was mentioned by you, Joanne, was the potential of kind of getting our name out there with insulin pump companies. We are in the stage right now where we're talking to pump companies about early stage research collaboration, partnerships for implementing this technology in our next phase of testing. And so whether it is your advocacy or advocacy of members of this group that could help us continue to drive the need for this type of technology, for them to want to pursue these types of opportunities with us, that would be so great. And we really cannot tell you how valuable that is for us. So on top of this data sharing, which is incredible, using your powers of advocacy for promoting this, I think would be a really, really great benefit that we could truly use. So I just really want to emphasize that.
But we're known for being really kind and really loud sometimes.
Yes.
So
if you're on Twitter, feel free to hashtag whatever you hashtag, especially we're not waiting, right? But we're on Twitter too. You can @ us at Diatec Diabetes. And that'll be really fun for us to see.
Yeah.
what
Every
might
time
be-
you have, oh, I was gonna say, every time you have a failure, if you want to take a picture, tag your respective pump company. Obviously, do it responsibly. We want to make sure that we tell them that this is something you want to see, but not something where we you don't want to kind of rub them the wrong way, I should say, right? We always want to be conscious of the fact that this is a sort of a professional environment, but we really think that if you guys believe in the technology and you want to see it come to happen, the work that you put into Night Scout, into Loop, into OpenAPS, that's kind of nudged the companies to do exactly what was already being done in the open source community. So if you say, we want to see this feature, we want to have Diatec be the ones to bring it to market, who knows, right?
Yeah, in this DIY looping community, we are either on Omnipod Eros pods or on old Medtronic pumps. There are others in Europe, but not here in the US. So I wanted to ask you, John, I thought I heard at the beginning you were on Tandem, but I'm catching on that you were on Omnipod.
Yeah, that's correct. Currently wear an Omnipod system. And so I am not a looper at the current moment, but I've been going back and forth between Tandem and Omnipod systems. But I've been using the pod for about, like I said, about 10 years. And I've seen a lot in my time as a podder, as I'm sure as everyone else has with their diabetes therapy. But yes, I can speak on behalf of experiencing infusion failures with this type of system.
So
And
you're on a Dexcom G6 as well?
currently I am, yes, I use a G6 intermittently. So I do not use the I'm very excited about the Omnipod 5 system. I think that that's going to be a really great great system for outside of the looping community. Of course, like I said, I'm not using Loop at the moment, but I've not really been too ingrained with the artificial pancreas system as of yet. So I'm moving closer to getting more so.
On this call, you've got some admins that are absolutely killer amazing at getting people up and running, and you have all the parts you need to loop. And wouldn't that be wise as a leader in your company to have experienced a closed loop before you fall back and do an FDA approved loop, which isn't out right now, that you'd really want?
Yes,
I don't know, any of my admins
yeah,
want to raise their hand and take them on?
Yes, I am all for the welcoming, and I think that this is really great to be able to meet with you all and have that potential opportunity. So I'm more than willing to talk with and try out. So
Okay, you admins, you got a new one, and it would be really interesting because then you would understand a whole bunch about the future and the critical importance to know what insulin's getting in. So we always look for converts. We actually don't. But you certainly are a prime candidate for it because you have the parts. As Tina says, it will rock your world, John.
that's awesome.
Anyway,
Yes, like I said, very excited about that type of opportunity. I've talked to Rayhan many a times about this type of technology. You know, I do have a good A1C. I haven't had one over 7 here in the last several years. But I think that, like, as you all have mentioned, there's so many things that can just disrupt everyday life with diabetes therapy, not just chronically, but acutely. And I think having this type of system really can allow for you to fix those little types of bugs and little types of issues.
well,
So
interestingly, I know Rayhan says over 7% is good enough. We don't quite think that way. And particularly the younger you are, the better we think you can do. And most, oh my gosh, one of the admins says we can do better than 7. And it's absolutely true. Probably most DIY loopers are down around 6 or below, and without effort, without a huge change. But I don't want to put you under the pressure. I just don't.
no, not at all. It's kind of funny. It's very ironic because my therapy is kind of old school to some of these things that we've been working with. So it's been really amazing to kind of get more so integrated with these types of technologies through Diatech. And so, like I said, bring it on. We're all for trying these new types of therapies to improve my own diabetes therapy, but of course others as well. So.
Oh my God, my admins are saying it will give you more time to bring us to better infusion sets. It'll get you to 5.5 with little effort. We're on your case.
I think we have a pool. I think we have a betting pool going for how many points you can lose. So looks like the spread is.
We can have another
1.5.
call and look at my
Your data. Yeah.
Oh my gosh, look at how much better this is. Yeah.
So, well, you know who would be the winner on this, but okay, I put you on the spot enough. Thank you so much for this. I think this surprised a lot of us, and it will be up and available, and I'll send you the link as soon as it's ready so that you can share it. I'll send you the chat, and we'll keep in touch because our people would love to send you some data and kind of move this forward because this is something we need. Absolutely.
Yeah, no, thank you so much for giving us the opportunity and the platform. We would love to work with your team, with your admins, or anybody who's in the community. I've actually seen a lot of it on Twitter, people from all over the world working on different modules for the different systems. And you know, we think that this can get done if it's not through the traditional business partnership, collaboration, whatever. I think we just go open source, right? And I think that there's enough capability here that we can do it together. And I think it's something that people want to see. And I think that you can't fight that. If this is something that's going to be helpful, we should make it available however we can.
We'd love to hear that. Listen, thank you. Everyone's saying thank you in the chat. I hope you guys have a really good evening, and we'll talk real soon. Thank you.
Thank you, everyone. I want to do a final disclosure. You know, this is all in development. We're happy with anything you share. But you know, what we have to say is that this is not a medical device that's for sale. Everything you share with us, we'll definitely use it to good effect, I hope, with development. But we won't be able to do any sort of active promises like that. But we appreciate the open source community, your spirit and your ethos. You are explicitly a little less hindered by the FDA as self-advocates. I think that's actually very admirable. But thank you for sharing this experience with us.
All right, JC, thank you. And thank you for that. We appreciate it. You have a good evening.